Good morning, everyone. Thank you for joining us today for our cancer testing frameworks. I'd like to introduce Patty Martin, the president and CEO of BioCrossroads. Patty, take it away.
Thank you so much, JoBeth. Good morning, everyone. We are so happy to have you here online. Still hoping that one of these days soon we'll be able to get together in person. But really, thank you so very much for participating in today's discussion, which is going to feature cancer diagnostics and screening. This is an upcoming emerging topic with lots of activity, and we have some great participants on the call today to share their knowledge and answer your questions. So to that point, I want to remind you to please put your questions in the chat. We're going to do our best to make sure that everything gets answered. And to kick it off, I want to thank our sponsor, Faegre Drinker. They are so supportive of BioCrossroads, and we are very thankful to have them supporting this educational series. So with that, I am going to kick it off and get started by introducing Hanny Mosley, Chief Medical Officer, Roche Diagnostics, North America. Hanny, it's all yours.
Thank you, Patty. Good morning, everyone. First of all, I would love to thank BioCrossroads for this kind invitation. It's a pleasure and an honor to be with this panel today, presenting to our great audience. I'm going to talk today about cancer diagnostics, and this is a near and dear topic to my heart. I have been with the Roche Group for almost 17 years now, mainly on the pharma side, but recently on the diagnostic side, and also mainly in oncology. Next, please. So maybe a little bit about Roche. I do not know if all of you know about Roche or not, but Roche is actually one of the oldest pharma companies in the world. It was actually established in 1986 by the Hoffman family, and it's based in Switzerland. Our headquarters for North America for Roche Diagnostics is actually based here in Indianapolis. And I'm proud to say that we have more than 3,300 people working every day to ensure the access of innovation to labs and physicians and also patients. We have been nominated as the #1 company in the field of R&D. We spend more than $13 billion every year for R&D, which is again reflecting our commitment to patients and healthcare systems around the world. We have been also nominated as one of the most sustainable pharmaceutical companies or pharmaceutical groups in the Dow Jones Index. And I'm really proud to be a member of this organization. Next, please. I wanted to start my discussion today with the patient journey because I think everything starts with the patient. And I think we are all doing this to ensure that we are improving patient outcomes, that we are having a positive impact to our patients and our societies. This is a picture of, I would say, the 2022 patient journey and how it looks. And when I look back to my career, when I started as a physician in Cairo, in Egypt, our patient journey was pretty simple. It was a patient presenting with some symptoms and signs. We diagnose, we treat, and there was not even a process of following up. And I think this patient journey has dramatically evolved in the next few years. Thank you to the evolution in cancer diagnostics and the introduction of new tests and technologies, and also the introduction of new therapeutics that actually making a huge difference in the life of our patients. I'm really excited to talk about some stages of this patient journey, especially the first probably 3 stages: screening, prevention, and early detection, because we are seeing great evidence now that actually early detection could lead to cure and will help us to manage cancer completely differently in the future. And also probably the last two or three stages, which is related to monitoring and disease management, which is also going to allow us to collect more real-world data to optimize the performance of our diagnostics and therapeutics and also inform future clinical research. Maybe I will share with you some statistics here. Actually, in the US, the number of women who have died of breast cancer has decreased by 41% from 1989 to 2018, thanks to early detection and treatment improvement. As a result, more than 400,000 breast cancer deaths were prevented during that period. So it's really important for us to understand this journey of evolution and how important it is this patient journey or our focus, moving more and more to early detection and prevention. I dream of an era where we can prevent cancer completely, or even deal with it as a chronic illness like any other chronic illness. Next, please. So this is a slide that we really love in Roche Diagnostics North America. And I have to say that diagnostics has historically been significantly undervalued, playing a foundational role in improving health outcomes. In fact, in vitro diagnostics impact 70% of healthcare decisions throughout the patient's journey. Yet the field accounts for less than 5% of healthcare spending in the US and only 2% global healthcare funding. I have to say that maybe the pandemic is changing or has changed this picture. And I think the value proposition of diagnostics industry generally is changing. The US spends more than $4 trillion every year on healthcare. 90% of this spend go to chronic illness and acute care. Less than 4% is spent on preventive care. Imagine if we can shift this equation. Imagine if we can actually spend more on preventive. Early diagnosis leads to better patient outcomes at a reduced cost. So you are not only improving patient outcomes, you are also helping this healthcare system to be more sustainable. Accurate screening, diagnosis, and monitoring can improve patient outcomes, helping them to spend less time in the hospital or avoid being hospitalized at all and enjoy a better quality of life. A wealth of information we can gather from a small blood or tissue sample is astonishing. And we are only just beginning to unlock the potential of this field. And I think Vince and Dr. Masoud will talk more about this in their presentations. Next, please. I would love also maybe to mention here some of the really interesting work we are doing at Roche and showing our commitment to cancer patients generally across the group. So one of the areas we are really focusing on is cervical cancer. And we are committed actually to eliminating cervical cancer and increasing access to solutions that can help improve patient outcomes. Approximately 92% of cervical cancers could be prevented by screening and vaccination against human papillomavirus, HPV. Regular screening can lessen the chances of cervical cancer developing. However, about 14,000 people in the US are diagnosed with cervical cancer each year and 4,000 deaths, according to the CDC. I'm really proud to work for a company which has a comprehensive cervical cancer screening, detection, and also a prognosis portfolio. Roche has advanced HPV and cervical screenings by pioneering tests that improve cancer risk assessment and confirmation. This can lead to finding cancer sooner, improving patient outcomes with earlier treatment. Roche is also helping clinicians to make better decisions through its FDA-approved portfolio that screens, triage, and diagnose for cervical cancer. These tools include the first and the only FDA-approved dual stain biomarker Syntic plus cytology to determine if a patient is progressing or not. I think also one of the most interesting areas that we are covering, and this is close to me because I also had the pleasure to work on checkpoint inhibitors and cancer immunotherapy in the US, lung cancer. And we all know that lung cancer is the leading cause of cancer death in the world, making up almost 25% of all cancer death. In the US, approximately 1 in every 15 men and 1 in every 17 women would be diagnosed with lung cancer in their lifetime. We are also very much committed to the lung cancer patient. And our commitment to lung cancer patients includes the development of more than 40 antibodies and genetic markers, including several that are FDA approved, to work in concert with several therapies used to treat lung cancers with the presence of PD-L1. One recent example is the PD-L1 or the SPE263 clone as a companion diagnostics for atezolizumab to help identify non-small cell lung cancer patients who could be eligible for immunotherapy. This is a terrific advancement in helping patients get an effective treatment earlier in the disease. one also of the areas that we are focusing on, and probably was my first area ever to touch in the field of pharmaceuticals, breast cancer. And Roche has been one of the leading companies actually in the field of breast cancer, developing our assays for HER2/neu overexpression. And we have helped millions of women in the last 20 years, either through our immunohistochemistry solutions or also through our pharmaceutical solutions through our monoclonal antibodies. We are also committed to all cancer patients. We have also some solutions for skin cancer, but these are probably the areas we are focusing on the most. I would love also maybe to mention precision medicine here, or maybe touch on NGS. And I know Dr. Masoud is going to have a great discussion about this, but we also have Foundation Medicine Corporation as one of the members of the Roche Group. And Foundation Medicine is primarily an NGS organization that's focusing on comprehensive gene profiling, and also working to actually advance liquid biopsies. And I think this is going to change the face of cancer care completely. So finally, if I want to summarize, I would just want to say that I feel that this is the golden age for cancer diagnostics. And the evolution we have been seeing in the last few years in that field is unprecedented, and it's actually helping us to fight cancer and to help improve the care of our patients. I'll stop here, and I'll be more than happy to answer all your questions at the end of this session. But let me introduce our next guest, my dear colleague, Vince Wong, Chief Commercial Officer for Geneoscopy. Vince.
Thank you so much, and great to be with you all here. And I am Vince Wong, Chief Commercial Officer with Geneoscopy. Next slide, please. Although I was a colleague with Dr. Masoud at Roche for 18 years, about 16 months ago, I jumped into the startup world and joined Geneoscopy, a venture-based startup out of St. Louis, serving as their Chief Commercial Officer. And our mission at Geneoscopy is to empower patients and providers to transform GI health through innovative diagnostics. We've heard a lot about precision medicine within oncology, and we're leveraging our RNA platform to transform GI health by bringing precision medicine into beyond oncology and into GI disease at large. We have a proprietary way of extracting and amplifying human RNA in stool, and are developing assays interrogating biomarkers to be able to manage GI health in a non-invasive way. Our first product is for colorectal cancer. Colorectal cancer is unfortunately the fourth leading cause of cancer in the United States. Over 150,000 Americans are diagnosed every year with colorectal cancer. And unfortunately, 1 in 3 Americans are not up to date with regard to their screening. And 60% of colorectal cancers could be prevented through screening. So our first product is to address this unmet need, and not only be able to detect colorectal cancer, but also high-risk precancerous polyps that over 90% of colorectal cancers start with a high-risk precancerous polyp, or otherwise known as advanced adenomas. And our first product is targeted to address the average risk population between the ages of 45 to 75. About a year ago, the American Cancer Society and the United States Preventative Services Task Force reduced the screening age for folks to get screened for colorectal cancer from 50 down to 45. So more and more people need to be screened due to the risk of colorectal cancer. We have a pipeline of products, and I'll talk a little bit more about that. Next slide, please. So the gold standard for colorectal cancer screening in the United States is to have a colonoscopy. Unfortunately, because of the unpleasant nature of colonoscopy, the invasive nature of it, requires people taking time off of work. It is obviously, the prep for a colonoscopy is not pleasant, and it requires sedation, and there are just a lot of barriers for people to get a colonoscopy. So what's really needed are accurate, non-invasive ways of doing colorectal cancer screening. So we are, leveraging our RNA platform to be able to interrogate RNA biomarkers in stool. And the reason why, specifically for stool, for colorectal cancer, stool is a better biological sample than blood due to the underlying biology and anatomy, where the stool passing through the colon will be able to capture cells that are shed in the colon, and we're able to interrogate those and be able to have a direct source of those epithelial cells. Whereas with blood, blood has a lot of promise for other types of cancer. It is less than an ideal sample type for colorectal cancer because it picks up a diffuse and distant signal, and you only get sensitivity for late stage disease as opposed to stage 1 or stage 2 or even precancerous polyps. And then, one of the things that attracted me to geneoscopy was at Roche, we salivated at the notion and the opportunity to be able to harness the power of RNA. But as most of you know, RNA is quite ephemeral and degrades quickly. What geneoscopy has developed is exciting technology to be able to harness the power of RNA, because it does provide a phenotypic and quantitative and dynamic snapshot of what's going on at the disease, whereas DNA, unfortunately, is more upstream and is genotypic and qualitative, providing limited insights into actual disease activity. Next slide, please. So how our test works is, if you are a sports fan like I am, you've seen the Cologuard commercials on TV, your ubiquitous ads. Our test has quite a similar patient experience, where the test would be ordered by a primary care provider. We would ship a kit to the patient's home. They send us back a sample to our CAP-CLIA lab in St. Louis. We would wash away the bacterial noise and extract and amplify and analyze the human RNA. We link the genetic features to the patient's phenotype, and then send the patient report to the ordering healthcare provider. Next slide, please. Colorectal cancer is one of several disease states we are developing across the continuum of care. We are developing screening assays for colorectal cancer, but also surveillance and recurrence, and also doing drug development in the area, but also looking at actively working with biopharma companies to develop assays in the areas of Crohn's disease, in ulcerative colitis, familial adenomatous polyposis, all areas of high unmet need. And so we're excited to be able to develop and ultimately launch a whole pipeline of tests to meet these unfortunately challenging diseases to manage. Next slide, please. So a little bit about where geneoscopy has been since its founding in 2017. It's a very cool story. Literally, company that started not literally out of a garage, but certainly in very humble beginnings by 3 young co-founders, and started with friends and family of $270,000 of funding. Last year we raised $110 million in our Series B and had a great set of investors, including LabCorp coming in as a strategic partner. We've gone from biomarker discovery and now happy to say we've just closed enrollment on our greater than 10,000 patient prospective clinical study, and we'll be filing with the FDA later this year. And we have breakthrough status, which will give us prioritized review with the agency. We now have 3 peer-reviewed publications and over 52 employees, and have moved from an incubator space in St. Louis into our own over 11,000 square foot CLIA laboratory and GMP manufacturing facility near the St. Louis airport. Next slide, please. Here's a snapshot of the 3 co-founders. That is not me. That's a much younger Dr. Kang. But in the middle is our scientific founder, Dr. Erica Barnell, who developed the technology while she was at WashU St. Louis School of Medicine. And interestingly, her brother to her right, who's the CEO, Andrew Barnell, when they were just young founders of the company. And now we've grown to, again, have 52 employees and on an exciting growth trajectory. Next slide, please. And we have been active with regard to, getting our data published. We're very proud of, having our data in peer-reviewed journals. Unfortunately, several of the other firms that are developing tests in colorectal cancer screening or multi-cancers early detection tests are lacking in peer-reviewed publications and prospective peer-reviewed publications, and we're proud of how we're going about generating robust data and getting that published in journals. Next slide, please. So our first study, our first prospective study that we published last year demonstrated that we not only can detect colorectal cancer with a high level of sensitivity, 95% sensitivity, but we're also, and probably the reason why the FDA gave us breakthrough designation despite the availability of Cologuard, is that we had a 40% improvement in advanced adenoma sensitivity over any other non-invasive test. So that's really what's needed and sought after within the GI community is a non-invasive way to not only detect cancer with a high level of sensitivity, but also to identify advanced adenomas, which again are those high-risk precancerous polyps that if you are able to identify those and have those removed, then you can actually prevent cancer, which at the end of the day is the whole point of cancer screening. Next slide, please. As I mentioned, the gastroenterology community is quite excited about the prospect of our test because of the promise it delivers and its opportunity to change the standard of care and moving colonoscopy away from being the first line screen in the United States for colorectal cancer. So as I mentioned, a 40% improvement over any other non-invasive tests in advanced adenoma sensitivity. We are filing again with the agency, FDA later this year, our CRC Prevent, our pivotal study. And, unfortunately, the pandemic has had a number of ill effects on prevention. 1.7 million colonoscopies were missed during the pandemic. And then 18,000 colorectal cancer diagnoses were either missed or delayed as a result. And unfortunately, there's a significant backlog for colonoscopies that causes there to be a greater need for non-invasive tests. And unfortunately, colorectal cancer, disproportionately impacts underserved populations. So 40% higher risk of colorectal cancer related deaths exists amongst African Americans. Unfortunately, colorectal cancer is not just a disease for the elderly. 1 in 5 colorectal cancer diagnoses will be for people between the ages of 20 to 54 years old. Again, we will read out our pivotal study later this summer, actually, and we'll be looking to launch our first assay by middle of next year, following FDA approval and CMS coverage. Next slide, please. So that's a little bit about genoscopy and colorectal cancer screening, and I'm pleased to be able to introduce my friend and colleague, Diana Caldwell, who is the CEO, president, and co-founder of Amplified Sciences. So Diana, take it away.
Oh, Diana, we cannot hear you. Sorry.
Can you hear me now?
Yes, perfectly. Thank you. Carry on.
Can you let me know if you can hear me now? No.
We can hear you, Diana. We cannot hear you.
You still
Sorry.
can't hear me.
There you
Diana,
are. There you are.
We
Can
can
you
hear
hear
you. So
Yes.
You can hear me now. Okay.
yes.
I clearly have had technical difficulties. I apologize. So do feel free to chime in if I leave you again. Great to be here, Vince. Thank you for overview of the great work you're doing, and I get the pleasure of working with Vince as he serves as a board advisor on our board. So 3 words upper left: accurate, earlier detection. I didn't put those on there for this program. That is what Amplified Sciences is all about. Next slide, please. So let me give you a quick company snapshot. We are birthed out of Purdue University. We were a Purdue Foundry startup. Dr. Joe Davison is the professor who invented our technology. He is a biochemist in the School of Pharmacy there. So we're an in vitro startup that's exclusively licensed some intellectual property from Purdue. And our mission is focused on accurately detecting and preempting the risk of debilitating diseases. What is our technology? We are a chemistry reagent platform, so we're not a box or an instrument, at least not yet. We're all focused right now on our very novel composition of matter reagent platform. And we're focused on building a portfolio of diagnostic assays for earlier disease detection that scales all the way to point of care. Our lead assay, and where we're starting actually, is in pancreatic cancer. We've heard about lung, colon. Pancreatic cancer is actually the 3rd deadliest cancer. It's a rare cancer, but because it's typically diagnosed too late, it has a very, very high mortality rate. And so we're focused on right now in pancreatic, additional areas of pancreatic, thyroid, and gastric cancers, and also more accurate earlier kidney disease detection. About funding to date, so we're very early, but we have raised $1.8 million in a capital raise, a seed round, closed in early 2021. And we've also earned some non-dilutive grants and just recently secured research use only revenue, which is just fabulous, in collaboration with other entities. Next slide, please. I'd like to talk first about the patient journey that's been spoken to earlier today, about what we're up to at Amplified Sciences. I want you to imagine for a moment that you are a patient and you've been having persistent stomach problems that are a little worrisome. So you book a primary care visit. She orders an abdominal CAT scan because she thinks it's probably your gallbladder. The CAT scan comes back clean for the gallbladder, but then she tells you she needs to send you to gastroenterologist. She says this is probably unrelated, but you have a growth, a cyst on your pancreas. You go home and do some Googling and you are scared. You realize that this could be pancreatic cancer, but may not be. And when you see the gastroenterologist, he tells you that you need to do more tests. We need to get more insight. We need to understand what's going on molecularly in that cyst. Next slide, please. Well, this is the first problem of many Amplified Science is working on solving. It's really the problem with growing incidental detection, meaning they're picking up cysts on the pancreas at an increasing rate. And those cysts are a window to earlier detection, which is what we've been talking about today. And you can't just ignore them because of the deadly nature of pancreatic cancer. 74 out of 100 patients are dead within the first year of diagnosis. The morbidity rate to remove those cysts is high. Type 2 diabetes, hospitalization, high reinfection rate. So you want to be sure that you understand and you can risk stratify patients, either no patients of concern, called non-mucinous, or patients that could be malignant or potentially malignant. Managing patients with pancreatic cysts that could have pancreatic cancer cost our health care system $4 billion. About 3 million patients a year are diagnosed with pancreatic cyst, and we know it's a window to earlier detection. And unfortunately for the patient, but fortunately for Amplified, the competition is plagued with both high false positives and high false negative rates. This all creates a huge unmet medical need, and it risks stratifying and managing these patients with pancreatic cyst. Next slide, please. Vince talked earlier about the importance of peer-reviewed publications and getting your data out there. So I'm proud to announce that our company became clinical stage in December of last year, December 2021. We had our first abstract poster presentation accepted that was submitted in February of this year. And we launched at the annual meeting our first clinical data. Don't expect you to able to see the details, but happy to provide more information if anyone's interested. We went head to head against the standard of care CEA utilizing pancreatic cyst fluid. Next slide, please. In terms of traction and where we are in our path, we've raised about $2 million to date, which I've mentioned. We are under IRB approval to collect samples. I'm proud to announce that IU is about to start our sample collection study with them. We're in great partnership with IU Simon Cancer Center and Dr. Al Haddad there. And we've secured some research use only, some other non-dilutive grant funding, and have 2 publications, one a poster presentation and then a full peer-reviewed and we've filed some new IP on top of our foundational Purdue IP, and we're quickly preparing for an assay that's ready to launch in a CLIA lab next year. Then we'll be ready to commercial launch. So rather than going 510K route first, we're going to go a lab developed test in a CLIA lab, which since COVID, many more of you have heard about those CLIA labs than ever before. In addition, we'll be building up our R&D group in order to scale additional assays in oncology primarily first, so that we have a portfolio of lab developed tests and 510K products in our own company, as well as searching out licensing technology. I want to spotlight the team, many of which have strong Indiana roots, as well as some new folks. So Dr. Joe Davidson, I've mentioned already, he is our inventor and my co-founder. Andy Cottrell, former Roche Diagnostics exec, is on our board as well. Dan Cheek, there in the middle, leads our R&D lab team, 2 full-times, as well as a part-time postdoc. Next slide, please. But we couldn't do it as a small startup of actually only 4 full-time employees without a great advisory board made up of clinicians, scientific talent, as well as surrounding ourselves with folks that have in vitro diagnostic launch experience. We've got Patty Connolly, that's a CLIA CLAP inspector, helping guide us on the regulatory process. We've got a dream team reimbursement team led by John Ridge, and also some business ops and back end office. And you'll see Vince there featured at top to the right, who's a very active entrepreneur in residence with the IU Ventures group that helps us tremendously. Next slide. As a startup, as you grow, one of the things is ensuring that you are earning the seat at the table, some of the larger diagnostic companies, and continuing to meet milestones to grow. That's what we're focused on. Surrounding ourselves with great clinical partners, other reputable academic institutions, entities like NIH and NSF. So we're pursuing all those opportunities, as well as being very active in the investor community, pitching our ideas as we prepare for our Series A. Thank you very much. Next slide. Anyone like to contact me? I think it'll be in the chat, but feel free to reach out. Next, I'm happy to present Dr. Ashik Masad. who's an associate professor of medicine at the IU School of Medicine, and will give us an oncologist perspective on this important topic.
Yeah, thank you, Diana. I appreciate it. So my colleagues did talk about diagnostic and early detection, but my role is more like an oncologist. Once patients are diagnosed, they come to our clinic. My interest and expertise are in cancer genomics. So I'll talk more about the role of cancer genomics. As an oncologist, I think this is the most exciting time to be an oncologist and take care of patients. Next slide. So the cancer we know for more than 100 years ago is a disease of genome. A scientist, Dr. Boveri, when he was studying sea urchins, he found under microscope that the large chromosomal translocations lead to these tumors in sea urchins. When he proposed his Boveri hypothesis, nobody believed him. But then after a few years, people started to believe him. And then the research started in the oncology world. People used to study smaller genes, etc. But the real watershed moment, in my opinion, was the completion of Human Genome Project. Next slide. That was completed in 2003, and it took more than a decade and few $1 billion, multiple institutions. Although it wasn't a complete architecture of a human genome, there were still a number of gaps which are still being worked up by a number of labs. But nevertheless, it was a huge, huge achievement in the field. Next slide. So why are we excited about Human Genome Project and understanding DNA, RNA expression levels? Because we know that the cancer is a genomic disease, and also it's a very dynamic process. So just to give an idea that on the extreme left, you see this as the embryo develops, it has its own intrinsic mutation in yellow, but as we grow, we are exposed to environmental factors, lifestyle exposures, etc. And then a benign tumor develops then into a cancer. Then you treat a patient with chemotherapy and the recurrence occur. So it's a very dynamic process, and it gives you a footprint how the tumor develops. This is one model to look at how the tumor develops over years. Next slide. But not only that, actually, if you look at this paper, this Minel paper, which was more than 10 years ago, this is a kidney cancer on the left. You see that these R1, R2 are the different sections which were biopsied. And then the tumor was studied, and it looked like the alterations, many of the alterations are shared, but many of the alterations in each spot were very unique, which tells us that the cancer is a very dynamic evolutionary process, and it evolves over time. And this is one of the roadblocks for the precision medicine, that which alterations to target as the tumors evolve, whether they evolve dynamically or under therapeutic pressure. Next. But then after Human Genome Project, I think this was the real time when people showed that we can do human tumor sequencing. And this was the first sequencing done in WashU in 2008 by Tim Lay, Rick Wilson, Elin Modis's group. And they showed for the first time that we can sequence tumors, and it was the first ever human genome. And I think it was a very smart move, because acute myeloid leukemia is relatively a simple, not at that time, genome to sequence and understand. Next. And what was more interesting around that time, their own colleague, Lucas Wortman, who is a lymphoma researcher, had acute lymphoblastic leukemia over years throughout his med school and fellowship, multiple recurrences. And behind you can see are these all sequencers in WashU, where he was sequenced because he had very few options, and not on his DNA, but on his expression profile, they found something called FLIT expression. He was treated with a drug which at that time was approved for kidney cancer, and he went into complete remission and went on to get a bone marrow transplant. And the last time I met him a few years ago, he had active lab. He was working on pro myelocytic leukemia. So at that time, it was still very difficult to sequence. They have to stop all other research projects, just focusing on their colleague. And a huge machinery of WashU started to analyze the data. Next step. Next slide. So at the same time, the Cancer Genome Atlas from NCI took this humongous task. As I was telling you, it took 13 to 15 years for Human Genome Project. They said that, can we sequence all tumors? As I said, that cancer is a disease of genome, it makes sense to sequence thousands of samples of different tumor types, not only the DNA, but also the expression profile, protein profiles, and more than 10,000 tumors have been sequenced thus far. So we have a large catalog which is publicly available for different groups to understand genomic biology at a much higher resolution than it was 20 years ago. Next slide. So one of the exciting things that is happening in the field is circulating tumor DNA. Next slide. So we know that as the tumors are growing, they shred these DNA into the bloodstream. The question, and we knew since 1960s that there are unique DNA from the tumor in the bloodstream, but the technology was not there, and it has really matured after the next generation sequencing. Next slide. So given that I'm a GI cancer doctor, I found this slide, which was very recently published, literally a couple of weeks ago. This paper talks about the power of next generation sequencing, because our other tests, including CT scans, PET scans, and other blood markers aren't as sensitive as circulating tumor DNA. On the left, you can see, you can draw a blood and try to understand these mutations and alterations at much high resolution, and try to figure out, is there anything targetable? The other important, which is a very exciting part of next generation sequencing in colorectal cancer, and is also evolving other tumor types, is minimal residual disease. Because after a patient had a surgery, you expect no tumor DNA in the blood, so you can actually customize unique alterations in the tumor, and then try to figure out if there is a DNA left in a bloodstream. If there is a tumor still in the bloodstream, that tells you there may be small cancer cells floating around that you cannot see on PET scan, CT scan. Next slide. On the extreme left, these are smaller studies from various groups that tells you that after surgical resection on early stage, for example, stage 1 and 3, these are the curable tumors. The risk is extremely high if a patient has circulating tumor DNA in the blood. And on the other slide, on table 2, you see many of these patients, when you give chemotherapy, you clear their DNA from this tumor DNA from the bloodstream. The question that is, are we really curing them with chemotherapy? And on the extreme right, a number of clinical trials are asking this question. Does ctDNA, can ctDNA guide therapeutics in colorectal cancer? Next slide. Now, what about the cancer care? It all looks fancy. We can do genomics whenever we want, but does it really matter when it comes to patient care? Next slide, please. So what is our goal when we are sequencing a tumor of a patient in our clinic? We want to benefit with minimal or no toxicity in our patients. That is the goal, to find a right drug to the right patient at the right time. Next. Okay, so these genomic data has really helped us to develop a number of therapeutics. For example, on the left, you see this drug vemurafenib. If somebody has this mutation called BRAF, now recently FDA has approved pan-cancer approval. If a patient has BRAF, these patients can get Herceptin. We all know has indication in breast cancer, gastric cancer, and even now in colorectal cancer, and also the gene signatures, which Hani earlier spoke about. Next slide. Next slide, please. Okay, so this tells you an example of a patient who relatively has a very aggressive anaplastic thyroid cancer, which is rare, was found to have this BRAF V600 mutation, was started on this drug with immunotherapy. On the left, you see these two dark lungs, and a central this white area, and you see after sustained response for more than 2 years on his post-treatment scan. Next slide, please. So one of the bottlenecks in genomic research is analyzing a data. It literally is finding a needle in a haystack. And then Indiana University has in my opinion, one of the best programs. Even before joining Indiana University, I came a couple of times to really look and try to translate this program at my previous institutions. So what is the Precision? A clinical program with an academic base, patients who have refractory tumor to standard of care options or have very minimal options, get cutting edge next generation sequencing of whole exome expression profile to identify options for these patients. And also not only that, the program has also committed genetic counselors to understand if they have predisposition to the cancer, and that will translate into teaching and reaching out to their family members, etc. And then the therapy selection, the way it goes, every week after a patient's history and tumors are sequenced, a group of experts, medical oncologists, biologists, bioinformaticians sit down together, go through the data, and try to come up with the best therapy options. Next slide. So one of the key strategy for success for this program was that it's not just only at the downtown location, but it was also expanded to other sites at IU locations. So patients will be benefited even though they may not come to Indianapolis location. Next slide, please. So how it works, a patient is referred to Precision Genomics program, history, physical exam, previous data is reviewed. This is followed by comprehensive cutting edge next generation sequencing and germline sequencing. Then the tumor board sits down together. It has group of oncologists who have expertise in different tumor types. As I said, and other colleagues come up with a plan. The options could be that to continue standard of care, early phase clinical trials, or sometimes non-standard FDA approved drug. Next slide, please. And also patient education is very key, and patients are educated throughout the process. Genetic counseling is done for their germline counseling. Not only that, one of the issue after next generation sequencing is to acquire a drug off FDA label, which program works very hard. And then also clinical trials matching, because it can be very overwhelming for our patients to find trials. So the program looks whether we have trial here or closer to home, or they have to travel at some other locations. Our program does best to match based on their location. And then we also follow up on their outcomes. Next slide, please. So here is an example. Some real world examples. So a 48-year-old female had esophageal cancer, first diagnosed early stage in April of 2019. This was followed by chemotherapy. Unfortunately, was found to have abdominal involvement. She was started on standard of care chemotherapy at that time, and then came to Precision Genomics, and her tumor was sequenced. Next slide. Now you can see she had this MLH1 mutation. Typically, it may not be picked up by the standard IHC testing, which is done in almost every patient. It appears to that she will, next slide, please. It appears to us that she will be a candidate for immunotherapy because of the DNA repair deficiency. Next slide. She was treated with immunotherapy Keytruda drug. Next slide, please. And here you can see on the left, a CT scan showed this omental or abdominal large mass, and in a few months, dramatic response to immunotherapy on this patient. Next slide, please. Another example of 62-year-old gentleman with metastatic pancreatic cancer. As we know, median overall survival is around a year now, started on standard chemotherapy of 3 chemotherapies called modified FOLFIRINOX, and was sent for precision genomics for sequencing, was identified to have a germline BRCA2 mutation, the same one that we see in breast cancer, is also prevalent in a subset of pancreatic cancer patients. Next. Again, it reaffirms that the patient had a BRCA2 germline mutation. Next slide, please. And then he was a candidate for a oral agent PARP inhibitor, and patient was put on this agent. Next slide. And there was a data confirming the benefit of this drug in germline BRCA mutated metastatic pancreatic cancer. Next slide, please. And you can see on the left that that was his pancreatic mass with a dramatic response after a few months of PARP inhibitor. Next slide. So in summary, next generation sequencing is revolutionizing care. And given the cancer is a disease of genome, oncology is a poster child for next generation sequencing. And it has really revolutionized the clinical care and will continue to do it. Technology is advancing at a higher rapid rate, not only for the treatment, also for minimal residual disease, to stratify patients who have much higher risk of recurrence and try to select those patients. Thank you very much.
Thank you so very much to all of our panelists, or all of our speakers that are now going to roll into being panelists. If you could please go ahead and put your camera and microphones on. Thank you so very much. So one of the things that we really like to do at BioCrossroads is to connect folks and continue to build out the network. And I know a number of you are already familiar with each other, but I'm wondering, and I'm going to start with Hani first, to just take, you heard your peers here. Is there anything, what did you learn today? Is there something that, or a point that you would want to make sure that's highlighted for our audience that would say, yeah, that's interesting?
So first of all, great presentations, and I learned a lot. But I think probably the highlight for me is that we are all here in the same journey, fighting to actually improve patient or have a better patient outcome. I really like what Dr. Masoud said about cancer being the disease of the genome. And I think the way you articulated this in a very eloquent way probably give us an idea what would be the future of cancer diagnostics and where we are heading with advances in NGS, with development of more probably liquid biopsy solutions, and also the ability to probably early detect and screen and hopefully prevent. So we are moving more to preventable diagnostics. So great presentations, and I was really happy to be in the panel today.
Vince, what about you?
I mean, similar to Hannah's comments, you know, it is truly exciting how, you know, to Hannah's points, I think we are in a golden era of diagnostics. I think you know, there's always silver linings to the pandemic. I think one of silver linings of the pandemic is it's highlighted the role and the criticality of diagnostics in the continuum of care, and has elevated the public awareness around things like that are important in diagnostics when we talk about test performance in terms of, you know, before the pandemic, no one knew what a false positive or a false negative was. That was a very foreign concept to the layperson. But now with COVID tests, we all know what, are all overnight experts on what test accuracy means. And I think that kind of fluency and awareness amongst the general public is going to be more and more important as diagnostics, especially in areas like oncology, that unfortunately, cancer touches so many of our lives, either directly or indirectly, to be able to have a better understanding of, what does this particular test tell me or not tell me, and you get a more informed and empowered a patient base as these technologies that we've all talked about become greater utilization and more ubiquitous and part of our standard of care. So I think that the talk really just highlights the breadth of technology and advances that in diagnostics that are transforming the patient journey in oncology overall.
It's interesting. I hadn't thought about that the whole term false positive, right? That's now more commonplace, as is emergency use authorization, which also had never really seen the lay press until. So there are silver linings. So we have questions from the audience. So rather than me taking up time, there's one for Dr. Masood, and it's around next generation sequencing and harnessing the power. I'm going to ask you a preliminary question, which is, how do you define next generation sequencing? And if that was covered, what exactly is that? What's old and what's new for a layperson?
Are you asking me?
Yes, sorry.
Okay. In the past, people used to sequence and study very smaller set of genes using Sanger sequencing and etc. They were kind of rough. Now it's a time when you take a genome and sequence it in days and have terabytes and terabytes of data coming out.
Okay.
And this is a massively sequencing data. And the technology is moving. Illumina has been at the forefront. I see Brandon's question, and now it's getting more and more interesting. Now we are moving into single cell technology, where you can understand a single cell of a tumor at the much higher resolution. I always give this example of single cell technology versus bulk technology, meaning a smoothie versus a salad. That is a bulk sequencing, and now we are moving into single cell technology. I see one of my colleague, Melissa Fischel, is in the audience, and I've been pushing single cell technology in our collaborations all the time.
Okay, that's great. So the specific question that was in the chat was, what do you see as the biggest challenge for harnessing the power of next generation sequencing related to tumor identification and characterization? What's getting in the way?
I think sequencing is much easy. Actually, taking a tumor and putting it in an Illumina machine is much easier. It's the analysis which is a bottleneck. I don't think what is happening, people do get reports, but to interpret the data and coming with the best possible option for the patients, I think is the real bottleneck. Okay,
that's great.
Yeah. And to follow on that, Dr. Masood, I think sequencing is very good with regard to biomarker discovery and identifying, a broad swath of potential mutations that might be happening for a particular patient. But, if we're able to then narrow for a particular disease state, the 8 or 9 or 12 biomarkers that are clinically relevant, and be able to translate that into, take the biomarker discovery that's being done at the NGS level, identify the subset of biomarkers that are relevant for a particular disease state, and then develop like a PCR panel that is able to interrogate those 8 to 10 biomarkers and allow for, that kind of panel to be run on a sustainable basis at a commercial lab level, that then generates clinically relevant answers, whether it be, for lung cancer, so there's probably 7 to 10 clinically relevant biomarkers in lung cancer right now where there's tangible treatments, FDA approved treatments that would be selected based on the particular biomarker signaling for that patient. So, that's how I see it as well.
Yeah, so the next question in here from Bob Marchesani is, how do you see these different value propositions working together? And Vince, you just suggested that you could end up with a panel of these, right? So is that, Diana, I'm going to ask you, is that something where pancreatic cancer is so rare, right? But unfortunately so very deadly. So where in the continuum do you see assays like yours working with other? I mean, do you see it being part of it? Well, it wouldn't be part of a panel because you're aspirating that particular tissue. But when you step back strategically and look at, wow, where could all of this go? Because I know you're thinking a few steps ahead. Where is that, and how does your technology, for example, fit in with others? And I'll come back to Hanny next and talk about how Roche sees this. Can't hear you, Diana. You're muted. No, you're not muted. I tell you what. I'm going to go to Hanny and ask him to unmute and talk about it, and then we can re-plug in and we can see what we can do.
Just checking. Am I back?
Oh, you're back. Okay, let's
Okay,
go to you
great.
while we've got you, and then we'll.
So on the fly, I'm diagnosing that I think I'm having a docking station go bad. So I think I've discovered it. So sorry if I think we know the root of my woes this morning.
Good to know.
Great question. And I think we need to think about the role of oncology diagnostics from screening tools. We've heard, maybe many of us, as we're tuned into this, the term liquid biopsy, where we're looking for evidence of cancer in more common, quite frankly, cheap to obtain samples, right? Blood, serum, et cetera. From screening to then, trying to give more of a prognosis and risk stratification. So how do we risk stratify those that we say, we might see something here? It could be a physical something like a growth, a cyst on a pancreas, or evidence in a blood sample, like a GRAIL, Thrive, et cetera. You guys have heard maybe some of these names because they've had big M&A activity in the last couple of years. So, and then risk stratification, and then therapeutic choices, really matching the best treatment path based on what the insights that either genomics are telling us or other biological indicators. We happen to be using an enzymatic activity biomarker for ours, where we're really looking at evidence of an active enzyme in activity, which is a little better than old residual evidence of some proteins, perhaps. But so we are not an NGS play at this point, right, but work together with them to potentially then look at genetic mutations that could be, for example, high-grade versus low-grade dysplasia with other biomarkers. So working in concert together, I think, along the continuum. And what I'm most excited about, my biggest takeaway, is making sure we're getting earlier. The role of diagnostics has been traditionally, something's gone wrong, right? And we need to help diagnose it. And I think where we're headed is both deeper into continuum of care, making the treatment the best, because we have smarter, better indicators of the specific patient, companion diagnostic revealing that allows us to pick that's later, and then pulling earlier, like where we're headed, which is trying to really identify potentially malignant or early malignant disease so that we can get ahead of it.
Okay.
I think I agree with everything that has been said. I liked a lot what Diana said. And maybe I would love to highlight here also the role of partnership between diagnostics or diagnostic companies and pharmaceutical companies or companies who are actually developing therapeutics. Because we know that when there is a certain therapeutic in an area, the diagnostic will be also accelerated. So I hope we can actually get more information, like pancreatic cancer is a great example. This is a rare cancer, but very hard to treat. And we do not have today really a good overall survival rate with most of the agents in the field. But imagine if we can work with a number of companies to accelerate research and development to provide a therapeutic. This is where we are going to see acceleration also in the field of diagnostics in pancreatic cancer. So I would love really to touch on this. We would love to see more partnership between diagnostics and pharmaceuticals generally.
Got it. Okay. So, Dr. Masood, the example that you gave of the pancreatic, of the two patients that you had that have come into the precision genomics program, I would be remiss if I did not highlight for you guys and our audience today that BioCrossroads is facilitating a large grant from the Lilly Endowment called Analytics Indiana. And that is focused on connecting genetic samples through the Indiana Biobank to clinical records so that we would have that continuum to study not just the genome, but also the clinical progression of a patient. And you mentioned the sites where you can collect the samples for the Precision Genomics Initiative. And I'm just a big fan of trying to understand how we can work with the Indiana Biobank, IU Health, the School of Medicine, to gather more samples, right? Because I think the more samples we have, the better off we're going to be. I don't have an answer for that right now, but I will tell you it's certainly something that we're very interested in, and we will be sequencing our first samples as a part of the Analytics Indiana project. So we'll come back and have another FrameWORX on that when we have actual data to share. But how comfortable are people, Ashik, with having their genetic information out there? I mean, is this something that you think people will be getting more comfortable with? Or are we still, is it big brother-ish? And I guess every person's going to be different, but where do you think the tide is turning? You're muted.
Sorry about that. I would say our patients are very kind individuals as long as they know that it will help somebody else down the line. I have rarely seen patients declining next generation sequencing. Patients are very kind, their families are very kind, and I don't think it's the major obstacle when we talk about next generation sequencing, in my opinion.
And that's generally patients who have been diagnosed with cancer already?
That's
Okay.
correct. That's correct.
Okay. All right, that's helpful. Hanny, anything from your point of view on this and getting, because the next place I'm going to go is to the payer perspective and how we think folks are going to feel about screening versus treatment. But
maybe one area that I think we should touch on or maybe discuss in more detail with, which is, and we didn't discuss it today, which is health equity and the access of probably our underserved communities to these technologies, because I think we need to do more in terms of education and supporting access to innovation in these underserved communities. So I hope we will need a different session, but I hope we can actually also try to fix this.
I can appreciate that very much. So Vince, I'm going to toss the last question to you and just ask, from a reimbursement perspective, right? Obviously, Cologuard, and now you have your FDA sanctioning of your uniqueness. Sorry, haven't had enough coffee today, but where are you thinking this is going with respect to reimbursement for screening?
Sure. Thanks for the question, Patty. So I used a metaphor of to describe the pathway from innovation at the bench to the patient, that companies have to slay a three-headed hydra in order to successfully get their discovery to the patient. So the first beast we got to slay is FDA and get approval from the FDA. Second is coverage from CMS, Center for Medicare and Medicaid Services, which covers about 30+% of U.S. population, the largest payer, typically 65 and above Americans. And then for screening tests in the areas of lung cancer, breast cancer, cervical cancer, and colon cancer, in order for a commercial payer, the Affordable Care Act mandates coverage of screening tests in those 4 solid tumor types if they are included in what's called the United States Preventative Services Task Force, USPSTF, which is a little known agency that sits nested under Health and Human services, but serves as a bit of a good housekeeping seal of approval for reviewing screening modalities and technologies that once you get approved within the USPSTF guideline, then commercial payers are mandated to cover that screening test. So, that's the pathway for any screening test within those four solid tumor types. Our test, because Cologuard is out there, does have a clear pathway for reimbursement, because the clinical utility of stool-based colorectal cancer screening is well accepted within the clinical community. And so payers will not treat our test as an experimental or investigational use device. And then, so there's three dimensions of getting paid. One is coverage, coding, and reimbursement. So from a coverage perspective, Cologuard's well covered, so we can ride that tailwind. From a coding perspective, we'll have to apply for our own CPT code. And then from a reimbursement perspective, there is our existing reimbursement rate, about $500 per test that Cologuard gets from CMS, that will likely be crosswalked in reference to. So long-winded answer to your question, but that's overall high-level pathway for diagnostics to get from the lab to the clinic.
I appreciate that. We do have one more question from Bob Marchesani, and then I'm going to turn it over to Lori Leroy. Do you guys have a response? He's asking about how diagnostic companies unapproved diagnostics can be partnered with pharmaceutical companies when they're not approved yet, right? So you can help with targeting populations, but that diagnostic isn't quite improved. So it sounds like a circle of life kind of thing, right? But any quick responses to Bob's question, and then I'm going to hand it over to Lori Leroy.
I can jump here. I don't think pharma or biotech are the main obstacle here. I think the real dialogue you need to have is actually with the agency, with FDA, and how this is going to help in the registration process, because this is where we face most of the challenges at the agency. So I would love to see a dialogue between pharma, biotech, diagnostics, and FDA to accelerate this innovation. This will be my comment.
Nothing's impossible for those that don't have to do it, right? So that sounds like an interesting conversation. So I'm going to thank our panelists and ask them to go ahead and first I want to really thank the panelists. So let me just take a pause here and say I really appreciate it. This is a topic that I'm sure will come back around and revisit it again as the technology progresses, and good luck to each of you in your endeavors. We love hearing about any kind of science that helps the great state of Indiana and patients as well. So if you don't mind turning off your cameras and muting your microphones, I'm going to do the same and hand it over to Lori Leroy, who's going to close us out.
Good morning, everyone. Thanks again for joining us and a fantastic discussion by our panelists. Thank you so much to Vince, Diana, Ashik, and Hani. Thank you for sharing your insights and perspectives. Looks like Indiana's got a lot of great work in this area, and there sound like there may be some great opportunities for partnership down the road. So again, thanks everyone for joining us. I just want to take a quick minute to once again thank our sponsor of this FrameWORX, Faegre Drinker. We greatly appreciate your support for this FrameWORX, as well as some of our upcoming events that I wanted to highlight real quickly before we end today. The first of which is our 23 Pair Award recognition ceremony. It will be on August 23rd. That's no mistake, 23 and 23. So we hope you guys will join us. It has been such a fun process in receiving the submissions and being able to notify some of the winners, which I will get to do again today. And so we encourage you to register on our website. Registration is currently open, and it will be a really wonderful event to highlight this next generation of life sciences leaders. It's so far a phenomenal list. It's going to be a lot of fun, and we hope that you will join us in person. So it will be our first, I think, in-person event that we've had in nearly 2.5 years. The other thing that I wanted to mention, just as a save the date, so registration is not open for the next event. However, we will have an event on, it's actually September 22nd of 2022. So mark your calendars. It'll be from about 2 till 6:30, 7 o'clock. We've got about 4 hours of really great content where we're going to share stories from the life sciences industry and BioCrossroads over the last 20 years, and then celebrate with a reception at the end of the afternoon. So we hope you will join us for that. Again, September 22nd. And we'll have registration up for that before too long. But again, an in-person event. So we've got 2 coming up here on August 23rd and September 22nd. We hope you'll join us for both of those. And we hope you have a great rest of the summer and look forward to seeing you hopefully in about a month in person. Thanks so much.